أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم فأين تذهبون إن هو إلا ذكر للعالمين لمن شاء منكم أن يستقيم وما تشاءون إلا أن يشاء الله رب العالمين صدق الله العلي العظيم. Lecture number 5, Gene Expression and Mutation as Sudden Change for second year class. Sudden change in genetic material is called as a mutation. I will start from simple information, then I will go to Mendel's law of segregation. What are the genes? First of all, the genes are the length of the DNA, and gene control, they are the parts. Genes are the parts of the DNA. Genes are the parts of the DNA and they control the character. Genes control character in the form of the pair, and in homologous chromosomes, how the genes are allocated, you can see. If the genes are same, it means if the genes are same, that is called as homozygous condition. If the genes are different in the homologous chromosome, the positions at which the genes are allocated on the chromosomes are loci, LOCI. So this is heterozygous condition, this is homozygous condition. One gene is dominant whose characters is appeared, and one gene is recessive whose character is not appeared. Main genetic material exchange during crossing over when two non-sister chromatids come closer at a point, chiasmata, you know. And gene pool, you know, the total amount of genetic material inside the nucleus of one individual, that is called as the genome. And genome is very important because genome carries the character. Genome of one individual consists of lot of genes. Those genes collectively called as gene pool. We talk about the gene pools. If you are talking about the gene pool, I will discuss one by one. The law of segregation, law of independent assortment. Gene pool is telling when the gene pairs are coming from the parents to the offspring, they will not bound with the other pair of the genes. For example, if you are taking two pair of genes simultaneously, in case of the law of independent assortment, I show you the crossing in which 9:3:3:1 was achieved. In the crossings of law of independent assortment, you will see two gene pair were used, two different gene pairs was used, capital R, capital R, capital Y, capital Y. This is the one gene pair. And what happens? Capital R, capital R, capital Y, phenotype is round yellow. I was talking about genotypes. When capital R, capital Y, this was the genotype, and round yellow was the phenotype. The mathematical result in law of segregation is a 3:1. If you talk about the ratios of the law of segregation, 3:1. Mathematical result of law of independent assortment is 9:3:3:1. But point I want to discuss, that point is much significant. The point which I just wanted to discuss, that is the significant. If you talk about capital R, capital Y, in this case, the phenotype is round yellow, mean Mendel crossed two different genes simultaneously in the same cross, unlikely that he did in law of segregation. So what happened in this crossing? In the F1 generation, dihybrid offspring is produced. What is a dihybrid? Two heterozygous conditions were produced. Capital R, small r, capital Y, small y, two heterozygous conditions were produced. Round yellow was the result. F1 generation, round yellow was the result. And this heterozygous condition was very good learning to make F2 generation, in which two options were there for capital R. Capital R has two probabilities, two chances in this case when it will go to form the gametes in for F2 generation. Capital R can go with small r and capital R can go with, definitely it can go with capital Y in this case, and it can go with the small y. So it has 50% chance capital R can go with the capital Y, 50% chance capital R will go with the small y. These two chances for the R, for the 50%, I will explain very easily by showing the characters in your body. Some characters in your body resemble the parents, those are the parental characters. Some characters in your body do not resemble your parents, those are non-parental characters. So if you see here the parents, capital R go with capital Y, it means that this was parental character. If the small r go with the small y, this was the parental character. If capital R go with the small y, this is the non-parental character. If small r go with the capital Y, this is the non-parental character. It means some characters during formation of the gamete remains like parent, and some characters, new characters are formed, recombination is formed. We can say mutation also, we can say these are the recombined genes, new characters which causes the variation. That was the achievement of the Mendel's law. He performed experiment in the pea plant. Scientific name of the pea plant was the Pisum stivum, is the Pisum stivum. So once again I will tell you this concept which is very important. I will love earlier this one, then I will write once again. Capital R, in this case, after the dihybrid mean two heterozygous conditions were formed. When one heterozygous condition is formed, that is called monohybrid. Capital R can go with capital Y, capital R can go with the small y. So it has the 50% probability, the 50% chance to go with the capital Y, 50% probability or 50% chances to go with the small y. In this case, similar case happens with the small r. Small r gene, capital R is the brother of small r, capital Y is the brother of small y, because they both belong to same family, but when they go in the gametes, they go with capital Y or small y, because in the gametes number of the genes becomes half, number of the chromosomes becomes half. Mendel wanted to tell us that each gene pair is independent of other gene pair. Each family is independent of another family. So capital R can go with capital Y, and capital R can go with the small y. Both chances are 50-50%. Both chances are there. These probabilities give strange result in F2 generation. What is the result in F2 generation? 9:3:3:1. I will discuss genotypically and phenotypically as well. Round yellow, if you see the parent which was taken in the parental cross, which were the true breeders, capital R, capital Y, these were the true breeders and they were the parental generations. What is the result in the crossings? Round yellow. So round yellow is basically parental character. Round green is non-parental character, because the capital R went with the small y. If you talk about wrinkled and yellow color, this is also non-parental generation. I write the genotype of wrinkled and yellow here. Look here, wrinkled yellow, wrinkled small r, wrinkled, and yellow capital Y. So this is also non-parental generation. I told you the reason why non-parental generations are produced. Now go back to fourth generation, wrinkled green. If I talk about wrinkled green, small r, small y, and wrinkled green generation. The wrinkled green generation, small r, small y, you can see this generation is found in the parent. Here is this generation. It was in the P1 generation, it was in the parental generation, it was the true breeder. So when the parents are combined, 50%, some characters resemble the parents, some characters do not resemble the parent. Reason, when more than one pair of the genes came into the gamete, what happens? They independently assort. Why they independently assort? During formation of gamete from F1 generation, you will get the idea. I will take this one and you will get the idea. I will just magnify then. Just hold on. I will magnify this one. Yes, I am able. Yes, I am able now. Look here. Capital R, small r, capital Y, small y. I can write here. Capital R, small r, capital Y, small y. So in the parental generation, look here. Capital R, small r, capital Y, small y. I want your attention. During formation of F2 generation, the gametes formation, capital R can go with capital Y, the one chance, one probability, one chance. Capital R can go with the small y, small r can go with the capital Y, small r can go with the small y. Now by taking this concept, I will make the gametes once again, and you will see, I will magnify this one once again. You will see capital R here in this case was having chance to go with capital Y. So that's why it become capital R, capital Y. Capital R has the chance to go with the small y. That's why it go with the it went with capital R, small y. Small r during formation of the gamete was having a chance to go with capital Y. So this chance was also availed by small r. Small r has the possibility to go with the small y. If I will rub this now, I will tell you which gametes are parental characters. Capital R, capital R, capital Y, they were the parental characters. Small r, small y, this is the parental character. These two are recombined genes. A recombination take place which causes the variation, which causes the mutation. Also you can say the change in genetic material is called mutation, and new characters are formed, due to which now coronavirus has become mutant. You know, now currently humans being became mutant because their characters are different from the past humans. Even animal bacteria has become mutant. After mutation, new characters are formed. You can see, I will tell you mathematically once again. Parental generation, I just wanted to show now. These were the parental generation: capital R, capital R, capital R, capital Y, round yellow, small r, small r, small y, wrinkled green. Now one of the generation I will assess, and you will just make a guess. You understand the recombined genotypes, the genotypes which are recombined. Look here, capital R, here this one. Capital R, capital Y, small y, round yellow, parental generation, of course. Small r, capital Y, small y, wrinkled yellow, no non-parental character, it is clear. Capital R, small y, round green, no non-parental character. So by this way the genes are expressed and the mutation take place. By crossing over also mutation take place. In the law of segregation, which was very simple, he used only one gene pair. You can see capital R was the round shaped seed of the pea plant, small r, wrinkled shape, and capital R was the gamete, small r was the gamete. They both combined and the generation was formed, capital R, small r, which was monohybrid. Monohybrid was self-crossed to form F2 generation, and F2, when it was self-crossed in F2 generation, when it was self-crossed in F2 generation, capital R, small r, multiply by capital R, small r. I am forming F2 generation. What will happen here? How many gametes will be, how many genotypes will be formed? Capital R gametes are shown by an circle. So 1 is the chance of the capital R. Now I can show you by here also, but before that I would like to tell you that 3:1 was formed in Mendel law of segregation. 3:1 was formed in the Mendel law of segregation. So these are the different contrasting character of the pea plant. I will draw the, yes, this is the 3:1 ratio I was drawing now. This was already drawn. So I was making this cross now some moment before. Capital R, small and capital R, small r. This was the result of F1 generation, mean it was monohybrid. When it was self-crossed, what happened? 3:1 was achieved. 3:1 mean round shape. This was round shape. This was also round shape. Small r present but dominant character will appear. Small r present but dominant character will appear. And small r recessive character 3:1 will be achieved. So Mendel law of segregation will show 3:1, but Mendel law of independent assortment actually shows the variation, some new characters. Test class already told in the classroom, when you want to identify the genotype of the dominant phenotype, this was little difficult task mathematically, but physically by doing experiment you can point out which genotype of which phenotype is present. Look here, we know round genotype, capital R, small r, but we don't know the genotype. Because genotype possibility is 2, capital R also round, capital R, small r also round. So you will check by crossing, by wrinkled, by recessive. So you will cross dominant generation with recessive generation. If one type of generation is produced, it means your genotype, both genes are dominant, like capital R in this case. Similarly, in the second experiment, if you cross the dominant once again with the recessive, the same experiment, you will do experiment one time, mathematically you will write 2 times. If the two generations are produced, it means your genotype is a heterozygous, capital R, small r. So these were some details about the genes expression. The genes exist in the form of the pair, and genes arrangement, genotype, their physical trait, phenotype, hybrid formed in F1 generation, monohybrid in F1 generation of the law of segregation. When more than 2 gene pairs are taken, the dihybrid is formed, and dihybrid is this one. And independent assortment give more data to us that how the genes express in the new characters, how the gene deviate from the arrangement of the parents, because some genes will not attain the same arrangement which they were having in the parents. So this is the dihybrid genotype, and phenotype will be in case of round yellow. So 4 gametes will be formed here, and 4 gametes will be formed here, and we will use the Punnett scale, and each gene pair will arrange in reference to their family. Mean capital R will go with the capital R, capital Y will go with capital Y. In the F2 of spring, in the gametes they will go in the different form. So these are the gametes in which I am talking about the probabilities of the chances that capital R can go with the capital R can go with the capital Y, capital R can go with the capital Y, and capital R can go with the small y. Similar in case of small r, and small r can go with capital Y, small r can go with the small y. These are the possibilities, and due to these possibilities new characters arise. Thank you very much.